Great afternoon, all. This is my research proposal. My name is Mika Martinez, and this is for class Sci C 750. The impact of cortisol on the brain, the release of cortisol due to psychological stressors. The purpose of this quasi-experimental study is to show the relationship between chronic psychological stress on the brain and the continuous release of cortisol and its effect on mental health related issues. I hypothesize that participants who are experiencing chronic psychological stress will be demonstrating higher cortisol levels compared to the control group, which has little to no psychological stressors in their workplace. All right, so in my study, there will be two variables. The participants in the control group will have a healthy workplace environment, will not have a formal psychiatric diagnosis, and will maintain a healthy work environment. For the dependent variable, that will have participants who are exposed to chronic psychological stressors in their work environment. And these participants will also not have a formal psychiatric diagnosis, and both participants will be engaging in the collection of the salivary cortisol samples. For the research design, participants must take salivary samples in the morning, at night, and throughout their workday. This is a quick and non-invasive method to measure biologically active unbound plasma levels of cortisol and allow saliva samples to be stored for further analysis. All right, so at this point, I'm sure everyone's kind of wondering who the participants are. I've chosen to narrow in on law enforcement as I'm a law enforcement officer, and we do experience stress on pretty much each tour. So for this research design, I have decided to do also a work stress diary with a specific section for them to note the date, the times, and the dates of their saliva samples that they collect. On those saliva sample vials, there will be a lot number and a vial number and a manufacturer number. And once they write down in their diary when they've taken the cortisol sample, they'll have to also have a documentation of that important information, as well as the date and time the sample was taken in order to ensure accurate tracking of the cortisol levels throughout the day. So each participant will be receiving 10 saliva vial samples per day for collection. Participants may not use all of them, but some days are heavier than others, meaning more things happen throughout the day for some officers, so they'll have more than enough to be able to, so we'll be able to measure their cortisol levels throughout their tour. In addition, if a stressful event occurs, they are to collect a sample of their saliva within 20 minutes and 40 minutes of the event in order to capture the immediate impact on cortisol levels. Again, that's what the 10 vials go for. Should they need more, they'll have. The saliva vials are designed to be safe at room temperature, which this allows the officers to be able to keep them with them during their shifts. All right, the methods section for the participants, data will be obtained from a small group of 20 male or female sworn law enforcement officers who are otherwise healthy and working in a small police department in North Carolina. Participants will also have at least a high school diploma, which as a law enforcement officer, you must have a minimum of a high school diploma. And the demographic characteristics such as race, ethnicity, age of the participants will reflect the diversity among the department. Therefore, we should have a range of backgrounds which will be present in the study. All right, the materials and procedure. Materials used will be, of course, unopened saliva vials, which will be kept in a designated white box for protection and organization. This white box will have two separate compartments, one for securely storing sealed vials and the other for safekeeping of the used vials. Used vials will be wrapped in clear plastic with, as stated before, serial, lot number, expiration date printed on it. And of course, the red journal will be attached on the top of the white box. Participants in this study will be briefed on the procedures to follow successful participation. This will include specific guidelines on when to draw the samples and how to handle the vials to maintain their integrity. For the procedure, the participants must wear sterile gloves provided in the box to prevent contamination. And I have a list here of chronological order of how to draw the sample and what steps to take. So first step number 1 is to record the date, time, and vial lot number, vial lot serial number in the journal provided. Number 2, to break the integrity seal on the swab and carefully remove the vial. Number 3, press the white collection paper against the inside of their cheek and drag it firmly towards the lips 2 times, making sure not to rub it back and forth in the cheek. To avoid contamination of the sample, they would need to push the slider cover up towards the tip of the collector without actually touching the sample that they put in their mouth. After collecting a sample, place it in the provided white envelope and seal it securely. And number 5, mark the date and time of the sample collection on the white envelope provided, as well as their full name and their date of birth. And that information is collected from the steps to this process is collected from the federal DNA database of a cheek swab. Lastly, participants in this study will not receive any form of compensation. However, their cooperation and willingness to participate will be sincerely appreciated. Thank you guys so much.